Hello everyone, exploring glucosinolates as biofumigants for control of soil-borne diseases. Glucosinolates are natural products in Brassica family that have the potential for use as biofumigants. More than 120 glucosinolate structures have been described, and most have cancer-preventing activities in human diet if consumed at low concentration. In plant production, biofumigation refers to the suppression of soil-borne organisms by compounds released into the soil. And the challenge in plant protection is to seek management practice that mitigate environmental pollution by pesticides while maintaining agricultural productivity. Broad spectrum of soil-borne diseases can cause severe damage to vegetable production. And what you can see here are symptoms caused by Phytophthora capsaicei. Glucosinolates in plants are broken down by enzyme known as myrosinase, and the products are isothiocyanate and mainly glucose and the thiocyanate and nitriles. The isothiocyanates are the active ingredient that cause the pest control. So glucosinolates themselves are not biologically active, but they must be enzymatically hydrolyzed by the enzyme to release isothiocyanate, the active ingredient. And when myrosinase contact glucosinolates in plant damaged or disrupted tissue, glucosinolates are released and the enzyme breaks it down into isothiocyanate. Now, metam sodium, or known as VAPAM, is a synthetic fungicide, and the active ingredient is isothiocyanate in this fungicide. But symptoms of poisonings are many. Some of them are eye irritation, reduction in the immune system, liver damage, and so on. But the difference with that and glucosinolates, the natural product, the active ingredient in glucosinolate is isothiocyanate, but they are slow released in the environment, and they are not sprayed but incorporated into the soil, so there is no drift and no damage by air. Our objective is to assess variation in glucosinolate concentration in turnip varieties grown under 7 soil management practices. These 7 management practices are sewage sludge, chicken manure, commercial organic fertilizer, commercial inorganic fertilizer, horse manure, and vermicompost. Now, we use 3 varieties of turnip, the purple top white globe and the Tokyo cross hybrid and the quartlet red stem turnip. Now, during the extraction process, we do not want the plant enzyme and the substrate to get together. So that's why we extracted the isothiocyanate using boiling methanol to inhibit the enzyme. As you can see also here that we developed a new method for the extraction of glucosinolates and determination of glucosinolates, as I mentioned, using boiling methanol, and then clean up of the plant extract using solid column, separation of glucosinolates using a Cefadex column, and the column here is disposable pipette tip, and then hydrolysis of the myrosinase, hydrolysis with the myrosinase on the pipette tip, and then quantification by determination of the glucose molecule. You can determine the glucosinolate by GC, by determining the isothiocyanate, and by determining the glucose moiety. As you can see here, we soaked the Cifadex in a solution saturated with ammonium acetate to load the ammonium ion onto on the Cifadex, and then add the plant extract, and then the plant extract will be bonded on the Cifadex, as you can see in the structure. There are many glucosinolates, as I mentioned. They are all differ in their structures, but they all have the isocyanate and the sugar moiety. So moles of glucose released into the aqueous media are equivalent to the moles of total glucosinolate in the plant extract. That's why we use the glucose to determine glucosinolates. As I mentioned, the GC can be also used, but takes a lot of time. Since every sample needs at least from 30 to 45 minutes, but with this method, we can use 100 of sample in 2 to 3 hours for that quantification. We used Cinegreen to fake a standard curve, and you can see Cinegreen is isocyanate, is a cycyanate that you release isocyanate. As you can see, the regression line is 0.95, which is great. So mydrogenase activity was measured by monitoring the hydrolysis product glucose using spectrophotometric determination. In conclusion, and regardless of the soil amendment, cultivar TC produced the highest concentration of glucosinolates, and we recommended this variety of turnip. Then regardless of the varieties, you can see that sewage sludge concentration, the concentration of the glucosinolates in sewage sludge amended soil was significantly similar to the commercial organic fertilizer that is used, and that's great compared to the other soil amendment, especially when you know that sewage sludge can be obtained almost at no cost. In conclusion, it is important for natural product to be water soluble. For potential field application, glucosinolates are water soluble. And then the enhancement of extracellular mydrogenase in soil amended with sewage sludge indicated that a lot of release of the glucosinolates was happening when plants were grown in sewage sludge. Soil amendments such as sewage sludge can be obtained free, as I mentioned. And actually, we recommend the sewage sludge for using for growing turnip and other brassica plants. If you also remember, some people are afraid about the use of sewage sludge to contain contaminant and bacteria, but class A biosolids that we used eliminate pathogen, including viruses. And remember that in the US alone, 6.5 million dry metric ton of biosolids were produced annually by the wastewater treatment plant. So they have a disposal problem, so that's why you can get it for free. These are the references that I use. I would like to thank the USDA National Center for funding this project under this agreement number. Thank you.